Amino acids are the building blocks of all proteins. In nature, there are more than 300 amino acids, however DNA codes for only 20 kinds of amino acids that are strung together in proteins. The core structure, for amino acids found in all proteins, have the same structure, including a central carbon, an amine group, a carboxyl group, a hydrogen and a R group. Only the R group varies in amino acid structure, that results in different amino acid structure. There are about 50% non-polar, hydrophobic amino acids, and the other half are polar, hydrophilic amino acids. Glycine is different from all the other amino acids, in having hydrogen on the R group, so having an H across from another H, making it the only kind of amino acid. Due to the hydrogen on both sides, glycine can exist in two mirror image forms, called stereoisomers, the L form and the D form. Thus is recognized as polar, and nonpolar uncharged amino acid. Chiral compounds that have different elements, molecules, attached to it in a solution, can make up 50% D forms and 50% the L form structure. Due to specificity of enzymes, it can only be recognized with producing only one form of the structure, either L form structures or the D form structures. However, amino acids made in cells, or used in protein synthesis, are mostly in L form. Amino acid is usually written in 3-letter codes, or 1-letter code. The 3-letter codes mostly contain first 3 letters of amino acids, however due to similarities in start letter, some adjustments are made. Non-polar amino acids include all those with alkyl chain R groups amino acids, the unusual cyclic structure amino acid, the sulfur-containing amino acid and aromatic amino acids. The alkyl chain R groups include alanine, valine, leucine, and isoleucine. Proline is similar to valine, but the R group of this amino acid loops around and bond to amino group, making an unusual cyclic structure. The one of the two sulfur-containing amino acid that is non-polar is methionine, and two aromatic amino acids are phenylalanine and tryptophan. Polar or hydrophilic amino acids are divided into 3 groups: the uncharged polar amino acids, acidic amino acids, and basic amino acids. The uncharged amino acids are usually more soluble in water than the nonpolar amino acids. The uncharged hydrophilic amino acids, containing good hydrogen bond-forming moieties, are the amide groups of asparagine and glutamine, the hydroxyl group serine and threonine, amphipathic group including tyrosine, and the sulfhydryl group of cysteine. The acidic amino acids contain carboxyls in their R groups, including aspartic acid and glutamic acid. Ionized forms of these are called aspartate and glutamate. The 3 basic amino acids, histidine and arginine, and an amphipathic amino acid, lysine, are called basic because their R groups accept protons at neutral pH, giving them positively charged R groups. The end with the free amino group is called the amino terminus or N-terminus. The other end is called the carboxyl terminus or C-terminus. The amino and carboxyl groups of amino acids can react in a head-to-tail fashion, eliminating a water molecule and forming a covalent amide linkage, which, in the case of peptides and proteins, is typically referred to as a peptide bond. When a few amino acids are joined in this fashion, the structure is called an oligopeptide. When many amino acids are joined, the product is called a polypeptide. The architecture of protein molecules is complex. Nevertheless, this complexity can be resolved by defining various levels of structural organization. There are as many as 4 levels of structure in a protein. The sequence of amino acids in a polypeptide is called the protein's primary structure. Proteins in any of their functional, folded conformations are called native proteins. The conformations existing under a given set of conditions are usually the ones that are thermodynamically the most stable. The change in protein structure is due to the amino acid structure and the interaction between them. The folding of protein occur, mostly as hydrogen bonds between atoms, in the peptide backbone, including carbonyl group of one amino acid and a nearby NH group of another amino acid. The 2 secondary structures identified are the alpha helix and beta sheet structures. In the alpha helix, the polypeptide backbone twists in a right-handed helix, such like the DNA. Every turn of the helix have almost 3.6 residues amino acids. In helix structure, the carbonyl oxygen of each residue forms a hydrogen bond, with the backbone of NH group 4 residues amino acid ahead. The beta sheet secondary structure consists of aligned strands of polypeptide, whose hydrogen bonding requirements are met by bonding between neighboring strands. The strands of a beta sheet can be arranged in parallel or anti-parallel beta sheet. Fibrous proteins are comprised mostly of primary and secondary structures, which sometimes combine into bundles, making long, strong fibers. Some fibrous proteins contain only a subset of the 20 amino acids that is organized in a regularly repeating pattern. Silk uses 3 amino acids to make beta sheets over and over, giving very high tensile strength. Collagen is the most important fibrous protein that holds cells together and makes up to 30% of the protein in humor. Non-fibrous proteins, the secondary structures features turns, which change the direction of the polypeptide chain, leading to complex three-dimensional structure. Proteins that fold into tertiary structures are called globular proteins, and they are the most abundant proteins in cells. In an aqueous cellular environment, polypeptides tend to fold, so that hydrophobic amino acids are buried in the interior of a globular protein. Cysteine, sulfur-containing amino acids, is the only amino acid that, with the covalent disulfide bond, significantly stabilizes the tertiary structure of protein. Proteins also interacts with metal ions. In contrast, a single metal ion can interact with multiple amino acids by coordination. The most common metals found in proteins are iron, zinc, and copper. Most small proteins consist of a single polypeptide chain, but most proteins with masses greater than 100 kilodalton contain multiple chains. Similar subunits of protein are known as homodimer, homotrimer, and homotetramer, in case the chains are not identical. Then the prefix is hetero, such as heterodimer and heterotrimer. The forces that hold subunits together are similar to those that determine the tertiary structures of the individual polypeptides. The spatial arrangement of these polypeptides is known as the protein's quaternary structure. There are two main bonding in protein, which when broken results in unfolding or denaturation of protein. These bonds are the disulfide bonds and the hydrogen bond. Disulfide bond is broken with mercaptoethanol, a chemical agent, and hydrogen bonds that can be broken using urea. After unfolding, the protein is no longer capable of its activity. Again, returning the unfold protein to folding position, or native position, is called renaturation, but sometimes cysteine misalign and forms wrong disulfide bond. Therefore, refolding, after denature of protein, is mostly unsuccessful. In the cell, there are small proteins called chaperones proteins, which assist protein in folding. Once folding has taken place, these chaperones are then no more associated with these proteins. A class of chaperones are also present, that misfolded dangerous or unwanted protein. Heat shock proteins, HSP, are molecular chaperones that refold partially heat denatured proteins in the cells. Chaperones also avoid association of hydrophobic regions of proteins with other hydrophobic component, which can lead to formation of toxic misfolded aggregates. Hemoglobin and myoglobin are globular proteins, however hemoglobin is oxygen carrier, and myoglobin is oxygen reservoir. Hemoglobin contains 4 polypeptide subunits, making it a tetramer, while myoglobin is a monomer. Hemoglobin is present in blood cells, however myoglobin is found in heart and skeletal muscle. When the oxygen level is very low, then myoglobin releases its oxygen. In hemoglobin, oxygen binds with iron, while carbon dioxide and proton, that are harmful for the body, is attached to the amino acid side chains, which is carried to the lungs, where they get released.